Welcome to the deep dive. Today we're really jumping into something crucial, a deep dive into biomolecules.
Absolutely.
And this isn't just, a standard chapter for your class 11th and 12th boards. It's actually vital for competitive tests like NEET and, well, other medical entrances too.
Extremely important, yes.
We always hear about all these chemicals and living things, right? From tiny bacteria up to, well, us. But what are they? How do scientists even figure them out? And maybe most importantly, what jobs do they do in our bodies? I'm really hoping we can unpack all of that today.
That's a perfect starting point. The living world, our biosphere, it shows this amazing, almost bewildering diversity, different shapes, functions.
Totally.
Yet, when you dig down to the fundamental chemical level, there's this profound, really fascinating similarity underneath it all. So our mission today is to systematically break down this chemical composition of living tissues. We need to differentiate the various biomolecules, really get a grip on their structures and functions, and understand how key processes work, especially enzyme action that drives almost everything.
Right, the enzymes.
And this isn't just about memorizing facts for exams, though it's definitely useful there. It's about grasping the essence of life's molecular machinery, stuff that's directly applicable to exam questions and real-world biology.
Makes sense.
So we'll start by looking at how living matter differs chemically from non-living stuff. Then we'll get into the analysis techniques, classify the biomolecules, and then dive deep into proteins, polysaccharides, nucleic acids, and critically, enzymes. They're just indispensable.
Okay, so let's tackle a really foundational question first, one that sort of sets the stage for understanding life chemically. Are living organisms actually made of the same basic chemicals as non-living matter, like rocks or soil?
Good question. I mean, intuitively, you'd think maybe not. Well, let's see. Imagine we do what's called an elemental analysis. We take a piece of living tissue, maybe a spinach leaf, a bit of liver, or even some microbial paste from a lab culture.
Okay.
And we meticulously identify all the elements present and their proportions. We'd find things like carbon, hydrogen, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon. You get the idea.
Right, a list of elements.
Now, what if we do the exact same analysis on a non-living sample? Let's say a chunk of the Earth's crust. What do we find?
The same elements?
Interestingly, yes. A remarkably similar list. In absolute terms, there isn't a single element found only in living things and nowhere else. Every element in the Earth's crust can also be found in living tissues.
Oh, okay. So that intuition was wrong then.
Well, hold on. Here's where it gets really interesting, and this is a crucial point for your exams. When you look closer, you see a significant defining difference. It's not what elements, but their relative abundance.
How much of each?
Exactly. Carbon and hydrogen, specifically, are present in dramatically higher proportions in any living organism compared to the Earth's crust. Let's look at some numbers. These are quite telling.
Okay.
Hydrogen makes up only about 0.14% of the Earth's crust. But in the human body, it's a huge 9.5%.
Wow, big difference.
Carbon is even more striking. It's just 0.03% in the crust, but a massive 18.5% in the human body.
18.5, how bad?
Oxygen is abundant in both, around 46.6% in the crust, 65.0% in humans, so it's high everywhere. But it's really the high concentrations of carbon and hydrogen that act as clear biochemical markers distinguishing living systems.
Right.
And conversely, look at silicon. It's a major component of the Earth's crust, 27.7%, but in the human body, it's negligible. Practically zero.
So lots in rocks, barely any in us.
Precisely. So while the types of elements are the same, the relative abundance, especially of carbon and hydrogen, is the true chemical hallmark of life. That's the key takeaway.
That's a really clear distinction. So it's not about unique elements, but the proportions, particularly that high carbon and hydrogen content. It's like a chemical signature for life. Makes sense.
Exactly, a signature.
So following from that, how do scientists go from just knowing the elements to identifying the specific organic compounds, like the actual molecules present in tissues? What's the method for that detailed breakdown?
Excellent question. And that leads us straight into the analytical techniques. Understanding this process step by step is really valuable, both for theory exams and practicals you might encounter. Okay, so to identify the organic compounds, scientists use a method that separates them into two main groups, or fractions. There's an acid-soluble pool and an acid-insoluble pool.
Acid-soluble and insoluble. Got it.
Right. First step, you take some living tissue. Could be simple, like a fresh vegetable leaf, or maybe a piece of animal tissue, like liver. That's your biological sample.
Right.
Next, you grind this tissue really, really thoroughly. You'd use a mortar and pestle, and you add a specific chemical solvent, trichloroacetic acid. Its formula is Cl3COH.
Trichloroacetic acid. Okay.
Yeah. The idea is to make a fine paste, like a thick, uniform slurry. You really need to break down the cells.
Right. Grind it up well.
Exactly. Once you have this slurry, the next critical step is filtration. You strain the whole mixture through something like cheesecloth or maybe a piece of fine cotton.
Like straining soup?
Sort of, yeah. This physical separation gives you your two fractions. The liquid that passes through the cloth, that's the filtrate. Technically, we call it the acid-soluble pool.
Acid-soluble pool.
And this pool contains thousands of different organic compounds. The key thing here is that these compounds generally have smaller molecular weights, typically in the range of, say, 18 up to about 800 daltons.
So the small stuff goes through.
Precisely. Now, what's left behind on the cheesecloth, the stuff that couldn't pass through, that's the retentate, also known as the acid-insoluble fraction.
Retentate, or acid-insoluble.
Right. And as the name suggests, this fraction contains the larger, more complex organic compounds, or simply those molecules that are insoluble in the trichloroacetic acid.
Like the big molecules.
Generally, yes. Once these two pools are separated, scientists then use more advanced techniques, things like chromatography, spectroscopy, to isolate and purify individual compounds from both pools. These methods help figure out their exact molecular formula and even their likely 3D structure. It's quite a process, breaking down something complex like a tissue into its individual chemical parts. Now, a general term, all the carbon-containing compounds we get from living tissues through analysis like this are broadly called biomolecules.
Okay, biomolecules.
But wait, living organisms also need crucial inorganic elements and compounds, right?
Like calcium, potassium.
Exactly. So how do we find those? We need a different approach. It's actually a destructive experiment.
Destructive, how so?
Well, here's how it works. First, you take a small amount of the living tissue, again, maybe a loaf or a piece of liver, and you weigh it carefully. This is its wet weight because it still has all its water.
Wet weight, got it.
Then you dry it completely, usually in an oven. All the water evaporates. The material that's left, that gives you the dry weight.
Okay, dry weight. Makes sense.
Next comes the destructive part. You take that dried tissue and you brain it completely, fully incinerate it.
Burn it, wow.
Yes. During burning, all the the organic molecules undergo oxidation. They turn into gases like carbon dioxide, CO2, and water vapor, and these gases are released.
So all the organic stuff is gone.
Exactly. What's left behind after everything carbon-based has been burned off and removed is just a powdery residue. We call it ash.
Ash, okay.
And this ash is precisely what contains the inorganic elements. If you analyze the ash, that's where you'll find the calcium, magnesium, potassium, sodium, and so on.
Ah, I see. So burning removes the organics, leaves the inorganics.
Precisely. Though it's important to note some small inorganic compounds like sulfates and phosphates are actually soluble and can also be found in that first acid-soluble pool along with the small organic molecules.
Okay, so they can be in both places sometimes.
Some of them, yes. So to sum up the analysis, elemental analysis gives you the list of elements, H, O, C, N, Cl, etc. Compound analysis, using the acid-dash methods, tells you about the specific organic molecules, like amino acids, sugars, and the inorganic constituents, like Na plus K plus say, K plus our, water, salts, phosphates, sulfates. From a chemistry viewpoint, we might look for functional groups, aldehydes, ketones, but biologically we classify them into categories that tell us about their function. Amino acids, nucleotides, fatty acids. That classification is more useful for understanding life.
That breakdown of the analysis really clears things up. Acid-soluble for small stuff, acid-insoluble for big stuff and lipids, and ash for inorganics. Got it. So focusing on that acid-soluble pool, you mentioned micromolecules like amino acids, fatty acids, and nucleotides. Could we maybe dig into each of those a bit more? I especially get confused between nucleosides and nucleotides. That difference always trips me up.
Absolutely. That's a very common point of confusion, and it's crucial for exams, so let's definitely clarify it. These micromolecules are, as you said, the fundamental building blocks. Understanding their basic structure is the key to understanding everything else. Let's start with amino acids. These are really unique organic compounds because they contain both an amino group -NH2 and an acidic carboxyl group, COH. And crucially, both groups are attached to the same carbon atom. We call this the alpha carbon,
Alpha carbon.
which is why they're often called alpha amino acids. You can think of them structurally as a central alpha carbon atom with 4 different things attached to it.
4 attachments.
Yes. One is always a hydrogen atom. One is the carboxyl group -COOH. One is the amino group -NH2. And the fourth one, this is the important one for variety, is a variable group designated as the R group.
The R group. So that's what makes them different.
Exactly. It's the chemical nature of this R group that gives each amino acid its distinct properties. While theoretically you could imagine tons of different amino acids, in living organisms we typically find only 20 specific types that are used to build proteins.
20 main types. Okay.
For example, in glycine, the simplest one, the R group is just another hydrogen atom. In alanine, the R group is a methyl group, NAFCH3. In serine, it's a hydroxymethyl group, CH2OH. You see how the R group varies?
Yeah, different little bits attached.
Right. And the overall properties, whether an amino acid behaves as acidic, basic, or neutral, depend on the nature of these groups, especially the R group, and whether it has extra amino or carboxyl groups. So you have acidic ones like glutamic acid, basic ones like lysine, and neutral ones like valine. Some also have aromatic rings in their R group, making them aromatic amino acids like tyrosine, phenylalanine, tryptophan.
Okay, so the R group dictates a lot.
It really does. And one more critical property often tested is the ionizable nature of the amino and carboxyl groups. Their charge can change depending on the pH of the solution they're in.
Ah, right. pH affects them.
Yes. And at a neutral pH, like inside our cells, they exist in a form called a zwitterion. ZWITTER ion.
Glucarion. Okay.
In this form, the amino group actually picks up a proton, becoming NH3+, and the carboxyl group loses a proton, becoming COO. So the molecule has both a positive and a negative charge simultaneously, but the overall net charge is neutral. This zwitterionic property is fundamental to how proteins fold and function. Okay, next up, lipids. What's the most defining thing about lipids?
They don't mix with water.
Exactly. General insolubility in water, that's key. This property makes them perfect for building cell membranes and storing energy efficiently. Let's look at fatty acids first. These are simpler lipids. Structure-wise, they have a carboxyl group, COOH, attached to an R group.
Another R group, like amino acids?
Sort of, yes, but here the R group is typically a long hydrocarbon chain. It can be quite short, maybe just a methyl, CH3, or ethyl, NACC2H5, or it can be much longer chains of NACC2H2 units, sometimes up to 19 carbons long, making 20 carbons in total with the carboxyl carbon.
Long chains.
Examples. Palmitic acid has 16 carbons in total. Arachidonic acid has 20 carbons. Now, these chains can be saturated. That means there are no carbon double bonds in the chain. It's saturated with hydrogen atoms. Think of solid fats like butter or lard.
Saturated fats, right?
Or they can be unsaturated. This means there's at least one or sometimes more carbon double bond, CC, in the chain. These double bonds introduce kinks. Think of most plant oils, like olive oil or sunflower oil, which are liquid at room temperature.
Unsaturated. Okay, double bonds.
Another simple but really important lipid is glycerol. Chemically, its name is trihydroxypropane.
Trihydroxypropane, which means 3 OH groups.
Exactly. It's a small 3-carbon molecule with 3 hydroxyl groups. This glycerol molecule often acts as a backbone for building more complex lipids.
A backbone, okay.
Many common lipids are formed when fatty acids chemically link to these hydroxyl groups on glycerol. This linking process is called esterification. Depending on how many fatty acids attach 1, 2, or 3, you get monoglycerides, diglycerides, or triglycerides.
Triglycerides, I've heard of those. That's like body fat.
Yes, triglycerides are the main form of stored fat in animals and also found in oils. They are what we commonly call fats and oils. The difference between a fat and an oil really just comes down to their melting point.
Melting point.
Yeah. Oils, like gingerly oil or soybean oil, have lower melting points, so they stay liquid at room temperature. Fats, like butter or ghee, have higher melting points, making them solid or semi-solid at room temperature.
Okay. Solid fats, liquid oils, based on melting.
Now, lipids can get more complex. Some incorporate phosphorus and a phosphorylated organic group. These are called phospholipids.
Phospholipids, key for membranes, right?
Absolutely critical. They form the basic structure of all cell membranes. Lecithin is a classic example you should know. And then certain tissues, especially neural tissues like your brain, contain lipids with even more intricate structures. The world of lipids is quite diverse.
Wow.
Okay, now let's tackle your point of confusion. Nitrogen bases, nucleosides, and nucleotides. Living organisms also contain carbon compounds that have rings containing nitrogen atoms. These are heterocyclic rings. These molecules are the nitrogenous bases, adenine A, guanine G, cytosine C, uracil U, and thymine T.
AGCUT, got it.
These are further classified. Adenine and guanine have a double ring structure, so they are called purines.
Purines, A and G double ring.
Correct. Cytosine, uracil, and thymine have a single ring structure, so they are called pyrimidines.
Pyrimidines. CUT single ring, got it.
Now, for the distinction you asked about, when one of these nitrogenous bases, A, G, C, T, or U, gets chemically attached to a sugar molecule, specifically a 5-carbon sugar, a pentose, like ribose or deoxyribose, the resulting molecule is called a nucleoside.
Base plus sugar nucleoside.
Okay. Exactly. So adenine plus ribose sugar gives you adenosine. Guanine plus ribose gives guanosine. Thymine plus deoxyribose gives thymidine. Uracil plus ribose gives uridine. Cytosine plus ribose gives cytidine. These are all nucleosides.
Adenosine, guanosine. Okay, those are nucleosides.
Now, if you take a nucleoside, which is base plus sugar, and you add a phosphate group, PO43, to it, specifically attached or esterified to the sugar part, then it becomes a nucleotide.
Okay, base plus sugar plus phosphate equals nucleotide. That's the difference.
That is precisely the difference. So a nucleotide is basically a phosphorylated nucleoside. Examples include adenylic acid, which is adenosine monophosphate or AMP, guanylic acid, GMP, uridylic acid, UMP, cytidyllic acid, CMP, and thymidyllic acid, TMP. These individual nucleotide units are the monomers, the building blocks.
The building blocks
for nucleic acids, like DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. These are long chains, polymers, formed exclusively by linking these nucleotide units together. And their primary, absolutely vital function is to serve as the genetic material, carrying all the hereditary information and directing protein synthesis. And just to round off the micromolecules in the acid-soluble pool, we also find simple sugars, which are technically carbohydrates. Things like glucose, C6H12O6, and ribose, C5H10OO5, are common examples. They serve as immediate energy sources for cells and also as structural components in other biomolecules, like the ribose and deoxyribose nucleic acids.
Right, glucose for energy. That was incredibly helpful, especially clarifying nucleosides versus nucleotides. Base plus sugar is the side, add phosphate and it's the tide.
Got it. So I see how these are the small building blocks found in that acid-soluble fraction. You also mentioned metabolites earlier, primary and secondary ones. Could you explain that distinction? And how does this connect to the macromolecules you mentioned, the big stuff in the acid-insoluble fraction? Great question, linking everything together. Yes, let's clarify the metabolites first. This distinction is actually quite interesting and highlights the diversity of life, especially in plants and microbes. So if we list all those biomolecules we just discussed, the amino acids, the sugars, the nucleotides, the fatty acids, these are fundamental to basically all life processes. We call these primary metabolites.
Primary metabolites.
But the key thing about primary metabolites is that they have clearly identifiable functions. We know what they do. They play known direct roles in the normal physiological processes of an organism, things essential for growth, development, reproduction. Glucose for energy, amino acids for proteins, nucleotides for DNA. These are all primary. You find them in pretty much all living cells, certainly in all animal tissues.
Essential for basic life functions.
Now, here's the twist. When scientists started analyzing plant cells, fungal cells, and microbial cells in detail, they found thousands of additional compounds besides these familiar primary metabolites. These other compounds are known as secondary metabolites.
Secondary metabolites, so like extra stuff.
Kind of, and this is a really important area for competitive exams, especially the examples. Think about pigments like carotenoids, make carrots orange, and anthocyanins, make flowers red or blue.
Okay, pigments.
Then you have alkaloids. Many of these are pharmacologically active, like morphine or codeine. Some are poisons.
Alkaloids, right.
Terpenoids, things like monoterpenes and diterpenes. These often contribute to the scents of plants, like in pine trees or citrus fruits. Essential oils, like lemongrass oil, are also in this category, valued for their aroma.
Smells and flavors.
Exactly. Then there are toxins, like abrin and ricin, which are potent poisons produced by some plants, likely for defense. Lectins, like concanavalin A, are proteins that bind specifically to carbohydrates. Then you have actual drugs derived from plants, like vinblastine, an anti-cancer drug, or curcumin from turmeric, and even polymeric substances like rubber, gums, and cellulose. Though cellulose is also structural, it's sometimes listed here in this context.
Wow, that's a really diverse list. Rubber, drugs, pigments.
It is. Now, the interesting thing about many secondary metabolites is that their precise role or function within the organism producing them isn't always clear. Unlike primary metabolites, their direct physiological necessity for the plant or microbe itself might not be immediately obvious.
So we don't always know why the plant makes them.
Often, we have hypotheses, maybe defense against insects, attracting pollinators, dealing with environmental stress, but their exact function isn't as universally defined as, say, glucose. However, what is incredibly clear is their usefulness to humans. We use them for rubber, drugs, spices, scents, pigments, and they definitely play important ecological roles interacting with other organisms. Okay, now let's connect this back to those acid-soluble and insoluble pools and the idea of biomacromolecules. Remember we said the compounds in the acid-soluble pool are generally small. Their molecular weights typically range from about 18 Daltons up to maybe around 800 Daltons.
Right, 18 to 800 Dalton macromolecules.
Exactly. We call these micromolecules or just biomolecules. Now, the acid-insoluble fraction, the stuff left on the cheesecloth, what did we say was in there?
Proteins, nucleic acids, polysaccharides, and lipids.
Perfect. Primarily those four types of organic compounds. Proteins, nucleic acids, polysaccharides, and lipids. Now here's the size distinction. With the very important exception of lipids, the other three classes, proteins, nucleic acids, polysaccharides, have much larger molecular weights. Generally 10,000 Daltons, 10,000 Dal, or even higher.
Wow, much bigger.
Much bigger. And for this reason, they are called macromolecules or biomacromolecules, macro meaning large. But this brings us to that critical exception, the point about lipids. This is a very common source of confusion and a favorite exam question. Why do lipids, which generally have molecular weights less than 800 Dal, putting them squarely in the micromolecule size range, end up in the acid-insoluble or macromolecular fraction?
Yeah, that seems contradictory. If they're small, shouldn't they be soluble?
It does seem contradictory, doesn't it? Let's break down the reason carefully, step by step. This requires understanding how cells are organized. Lipids are small molecular weight compounds, that's true. But inside a living cell, where are they primarily found? Are they just floating around dissolved in the cytoplasm?
No. They make up membranes, right? Cell membrane, organelle membrane.
Exactly. They are primarily organized into complex structures, most importantly the cell membrane and the membranes of various organelles like the endoplasmic reticulum, Golgi apparatus, mitochondria. They form the fundamental bilayer structure of these membranes.
Okay, part of membranes.
Now remember what we do in the first step of the analysis. We take the tissue and grind it vigorously in trichloroacetic acid.
Right, the grinding step.
What does that grinding do to the cell structure?
It breaks everything apart, shatters the cells.
Precisely. It disrupts and breaks down the entire cell structure. The cell membranes and all those other internal lipid-rich membranes get physically shattered into tiny fragments.
Yeah, membrane pieces.
Now, what happens to these little pieces of oily, fatty membrane when they're suspended in the watery trichloroacetic acid solution? Do they dissolve?
No, oil and water don't mix. They stay separate.
Exactly. They don't dissolve. Instead, these broken membrane fragments tend to spontaneously form small, closed, insoluble structures called vesicles, little bubbles of membrane.
Vesicles, okay.
Because these lipid vesicles are insoluble in the acid, and because they are physically larger entities than individual dissolved micromolecules like glucose or amino acids, they get trapped along with the true large macromolecules, proteins, nucleic acids, polysaccharides, in the acid-insoluble pool when we do the straining step.
So it's not their individual molecule size, but the fact that they clump together into these insoluble vesicles when the cell breaks.
That is the perfect way to put it. So to be absolutely crystal clear for your exams, lipids are not strictly macromolecules in the sense of being huge polymers like proteins, but they get separated with the macromolecular fraction because of their role in membranes and their resulting insolubility and aggregation into vesicles during the extraction process. It's a consequence of the analysis method and their cellular organization.
Okay, that makes so much more sense now. It's about their behavior during extraction, not just their size.
Right. So recapping the fractions. The acid-soluble pool roughly represents the cell's cytoplasm composition, all the dissolved small molecules. The macromolecules, proteins, polysaccharides, nucleic acids, from the cytoplasm and organelles, Plus, those lipid vesicles form the acid-insoluble fraction. Together, these two fractions give us the entire chemical inventory of a living tissue.
The whole picture.
Exactly. And just to put in perspective, remember the overall composition? What's the most abundant chemical in a cell by weight? Water. By far. Water makes up a massive 70–90% of the total cellular mass. After water, proteins are the next most abundant, around 10–15%. Then nucleic acids at 5–7%, carbohydrates around 3%, lipids about 2%, and finally inorganic ions making up about 1%. Knowing these relative percentages is also important.
Okay, that lipid explanation was key. So we've got the small stuff, micromolecules, and the big stuff, macromolecules, including lipids by association. Let's now really dive into those true macromolecules, proteins, polysaccharides, and nucleic acids. Proteins especially, you said they're the workhorses. They seem to do almost everything. How do they achieve such amazing versatility?
Indeed, their versatility is astounding, and it all comes down to their structure. Let's start with them. Proteins, fundamentally, are polypeptides. What does that mean? Poly means many. Peptide refers to the bond-linking amino acids. So proteins are long, usually linear chains built from individual amino acid units linked one after another by a specific type of chemical bond called a peptide bond.
Peptide bond, okay.
This bond forms through a condensation reaction. A molecule of water is removed when two amino acids join together. Now, here's a crucial point. Each protein is a polymer made of amino acid monomers. How many different types of amino acids are commonly used to build proteins?
20, you said earlier.
Exactly, 20 different types, alanine, cysteine, lysine, etc. Because proteins are built from a variety of these 20 monomers, they are always heteropolymers. Hetero meaning different.
Heteropolymer, okay, made of different units.
Right. This is a key distinction. A homopolymer, in contrast, would be made of only one type of monomer repeating over and over. Like if you had a chain made only of glucose units, that would be a homopolymer. Proteins are never homopolymers. They need the diversity provided by those 20 different amino acid R groups to create complex, three-dimensional structures and specific active sites required for their vast range of functions.
That variety is essential for their function.
And thinking about those amino acids brings us to nutrition. You've probably heard of essential amino acids.
Yeah, the ones you have to get from food?
Exactly. Essential means our body cannot synthesize them itself. We must obtain them from the proteins in our diet. Non-essential amino acids, on the other hand, are the ones our body can make from other molecules, so we don't strictly need them in our diet. Now, the functions. Proteins perform an incredible array of functions. This list is prime material for NEET and board exams. Questions about specific protein roles are very common. Let's look at some key examples. Collagen. Remember this one. It's the most abundant protein in the animal world. What does it do? It provides intercellular ground substance, basically acting like the structural scaffolding or glue in connective tissues, tendons, cartilage, bone matrix.
Collagen. Structural protein, got it.
Trypsin. This is a classic example of an enzyme. Its job is to catalyze specific biochemical reactions, in this case, breaking down proteins during digestion.
Trypsin, an enzyme.
Insulin.
Yeah.
This is a hormone. It's a chemical messenger that plays a critical role in regulating blood sugar levels.
Insulin,
Yeah.
hormone.
Antibody. Antibodies are proteins that are key players in our immune system. Their function is to fight infectious agents like bacteria and viruses.
Antibody fights infection.
Receptor proteins. These are usually found on cell surfaces and are involved in sensory reception. They detect signals from outside the cell, things like smells, tastes, or the binding of hormones or neurotransmitters.
Receptors receive signals.
GLUT4. This is a specific example of a transporter protein. Its job is to enable glucose transport into cells, which is vital for getting energy into the cell.
GLUT4 transports glucose.
Okay. And two really important facts about abundance, often asked in competitive exams. Collagen is the most abundant protein in the animal world, but ribulose bisphosphate carboxylase oxygenase, usually just called Rubisco.
Rubisco, that sounds familiar from photosynthesis.
Exactly. Rubisco is the enzyme that captures carbon dioxide in photosynthesis. Because photosynthesis is so widespread, Rubisco is considered the most abundant protein in the entire biosphere.
Wow. Most abundant overall.
Rubisco. Okay, moving on to the next class of macromolecules. Polysaccharides. These are essentially long chains of sugars, so they belong to the broader category of carbohydrates. They are also found in that acid-insoluble pellet. Think of them literally like a cotton thread, but instead of cotton fibers, it's built from monosaccharide, simple sugar, building blocks linked together.
Long chains of sugars.
Okay. Many important polysaccharides are homopolymers, meaning
Made of only one type of repeating unit.
perfect, made of just one type of monosaccharide. Cellulose is a prime example. It's a polymer consisting only of glucose units linked together in a specific way. It's a homopolymer. Where do we find cellulose?
Plant cell walls, paper, cotton.
Exactly. Plant cell walls, paper, cotton fiber. Structurally, cellulose forms long, straight, linear chains. An important point for lab tests. These linear chains cannot easily trap iodine molecules. That's why cellulose doesn't give a blue color with iodine solution.
No blue color for cellulose. Starch is another very important glucose polymer. It's the main way plants store energy, like in potatoes or rice. Now, starch is also made of glucose, but the glucose units are linked differently than in cellulose, causing the chains to form helical or coiled secondary structures. Starch is coiled.
Yes, it forms helices. And this is crucial because these helical portions can trap iodine molecules. That's why adding iodine to starch results in that characteristic blue-black color, a very common test you've probably done in school labs.
Ah, the helix traps the iodine. That's why starch turns blue. Okay, glycogen is often called animal starch. It's the way animals, including us, store glucose for energy, mainly in the liver and muscles. It's also a polymer of glucose, but it's much more highly branched than starch. Think of a main chain with lots of side chains coming off. Glycogen, animal storage, branched. Got it. Referencing figure 9.2 mentally. Inulin. Just another example to know this one is a homopolymer specifically made of fructose units, not glucose. Inulin, polymer of fructose.
In a polysaccharide chain like glycogen or starch, the two ends of the chain are chemically different. One end is called the reducing end, and the other is the non-reducing end. This difference is due to the way the sugar units are linked.
Reducing and non-reducing ends.
Now, besides these relatively simple homopolymers, nature also makes more complex polysaccharides. These often use modified sugar units as their building blocks. For example, sugars with an amino group attached, amino sugars like glucosamine, or other chemically modified sugars like N-acetylgalactosamine. A very important example of such a complex polysaccharide is chitin.
Chitin, like in insect shells.
Exactly. Chitin forms the hard exoskeletons of arthropods, insects, spiders, crustaceans, and it's also found in the cell walls of fungi. Even though it uses modified units, N-acetylglucosamine, chitin is also considered a homopolymer because it's made of just that one type of repeating modified sugar unit.
Chitin, exoskeletons, fungi. Still a homopolymer.
Okay.
Yeah.
All right, the final major type of macromolecule found in that acid-insoluble fraction, nucleic acids. As we established earlier, these are polynucleotides. Together with polysaccharides and polypeptides, proteins, these nucleic acids make up the bulk of the true macromolecular fraction of any living tissue or cell.
Polynucleotides. Right.
And what's the fundamental building block, the monomer, of a nucleic acid?
The nucleotide. Base plus sugar plus phosphate.
Perfect. The nucleotide. And just to quickly refresh, a nucleotide has those three distinct chemical parts. The heterocyclic nitrogenous base, AGCT, the monosaccharide sugar, a pentose, and the phosphoric acid or phosphate group. Remember the bases. Purines, AG double ring, and pyrimidines, CUT single ring.
Yep.
And the sugar in polynucleotides is either ribose, the pentose found in RNA, or 2-deoxyribose, the pentose in DNA, which lacks an oxygen atom at the 2' carbon position.
Ribose in RNA, deoxyribose in DNA.
Correct. So naturally, a nucleic acid containing deoxyribose is called deoxyribonucleic acid, DNA. And one containing ribose is ribonucleic acid, RNA. And their primary function, the reason they're so central to life—
Genetic material, carrying information.
Exactly. They function as the genetic material, carrying hereditary information from generation to generation and directing the synthesis of all the proteins a cell needs to function.
That's a great overview of the three main types of macromolecules: proteins, polysaccharides, nucleic acids, built from amino acids, sugars, and nucleotides, respectively. It's becoming much clearer how these large molecules are constructed. Now, going back to proteins for a moment, I remember hearing about different levels of protein structure, like primary, secondary, tertiary, even quaternary. How do biologists define these levels, and why is understanding them so critical for a protein's function?
Excellent question. Yes, the concept of structure for proteins is hierarchical, and understanding these levels is absolutely crucial because the final 3D structure dictates the protein's function. Biologists describe protein structure at 4 distinct levels, and you really need to grasp each one for your exams. Let's visualize this as we go, mentally referencing figure 9.3. The first level is primary structure, 1 degrees. This is the most fundamental level. It simply refers to the sequence of amino acids in the polypeptide chain.
Just the order of amino acids.
Exactly. Which amino acid comes first, which is second, third, and so on, right to the very end of the chain. Think of it like the specific sequence of letters that make up a unique word or sentence.
Okay, the linear sequence.
Yes. And we represent this linear chain with a starting point and an end point. The beginning, the first amino acid, is called the N-terminal amino acid because it has a free amino NH2 group. The end of the chain, the last amino acid, is the C-terminal amino acid with a free carboxyl COOH group.
N-terminus and C-terminus, got it.
The importance of primary structure cannot be stressed enough. This linear sequence, which is directly determined by the genetic code, DNA, dictates everything else about the protein's folding and its ultimate function. Even changing just one amino acid in this sequence can sometimes drastically alter the protein's shape and make it non-functional. Think of sickle cell anemia caused by a single amino acid change in hemoglobin.
Wow, just one change can do that.
Okay, next level, secondary structure, 2 degrees. The polypeptide chain doesn't just stay as a long, straight string. Parts of the chain start to fold and coil locally into regular, repeating patterns.
Folding starts here.
Yes, localized folding. This folding is driven mainly by hydrogen bonds forming between the CO group of one peptide bond and the NH group of another peptide bond along the polypeptide backbone. Importantly, the R groups are not directly involved in forming secondary structure.
Okay, backbone interactions.
Right. There are 2 major types of secondary structure you need to know. The alpha helix. A helix. This is a common pattern where the backbone coils into a helical shape, like a spiral staircase. An interesting detail. In proteins found in nature, these are always right-handed helices.
Alpha helix, like a spring.
Yeah.
Right-handed only. The beta-pleated sheet, pleated sheet. In other regions, the polypeptide chain folds back on itself or aligns side by side with other segments, forming a flattened zigzag sheet-like structure.
Yeah.
This is also stabilized by hydrogen bonds between backbone atoms on adjacent strands. Beta sheet, like folded paper.
Okay. These secondary structures, helices and sheets, add stability and provide the basic motifs that will fold further. Now for the tertiary structure, 3 degrees, this is where the real complexity comes in. The entire polypeptide chain, including its alpha helices and beta sheets, folds and bends upon itself extensively to create a unique, complex, 3-dimensional shape.
So the secondary structures fold up further.
Exactly. Imagine taking that coiled spring, alpha helix, and those folded sheets, beta sheets, and then further crumpling and folding the whole thing into a specific, compact, globular shape, maybe like a tangled ball of wool, but a very specific tangle.
A complex 3D shape.
Yes, a very specific 3D shape. And this structure is stabilized by various interactions between the R groups of the amino acids. These interactions include hydrogen bonds between R groups, disulfide bonds, strong covalent bonds between 2 cysteine R groups, ionic bonds between charged R groups, and hydrophobic interactions, where nonpolar R groups cluster together away from water.
Interactions between the R groups hold it together.
Precisely. And this specific tertiary structure is absolutely essential for the biological activity of the protein. It's this precise 3D shape that creates the functional sites, like the active site pocket in an enzyme, where the chemical reaction happens. If a protein loses its tertiary structure, denatures, it usually loses its function.
Tertiary structure
Same,
and motion.
yeah.
Got it.
Finally, there's the quaternary structure, 4 degrees. This level doesn't apply to all proteins. It only exists for proteins that are composed of more than one polypeptide chain. Each individual polypeptide chain in such a protein is called a subunit.
So multiple chains working together.
Exactly. Each subunit has its own primary, secondary, and tertiary structure. The quaternary structure describes how these individual folded subunits are arranged and interact with each other in the final protein complex.
How the different pieces fit together.
Perfectly put. The arrangement could be simple, like 2 identical subunits paired up, or more complex, like several different subunits arranged in a specific geometry, maybe like a cube or a ring. A classic example you absolutely must know is adult human hemoglobin, HB.
Hemoglobin for oxygen transport.
Right. Hemoglobin is made of 4 subunits, 2 identical alpha subunits and 2 identical beta subunits. The precise way these 4 chains fit together defines its quaternary structure, and this specific arrangement is crucial for its ability to bind and release oxygen efficiently. Many complex biological functions rely on these multi-subunit quaternary structures.
4 levels. Sequence, local folds, overall 3D shape, and subunit arrangement. Okay, that clarifies the hierarchy. Wow, protein structure is way more intricate than I realized. Those 4 levels build up so much complexity. And you mentioned enzymes are mostly proteins, which, yeah, That's a huge topic for NEET and boards. Can we really dive deep into how they work? What makes them speed up reactions so much and what things affect how well they work? Especially that concept of activation energy that's always seemed a bit abstract and tricky to me.
You're absolutely right to focus on enzymes. They are a cornerstone of biochemistry, and yes, a favorite topic for exam setters. It requires careful attention, so let's take our time and go through it systematically. First, just a recap on their nature. As we've established, almost all enzymes are proteins. They possess those primary, secondary, and tertiary structures. Now remember that active site we talked about in the tertiary structure? That specific crevice or pocket on the enzyme surface is where the substrate binds and the catalysis actually happens.
The active site. Key location.
Absolutely. Now, there is that small exception. Some RNA molecules can also act as catalysts. These are called ribozymes. But the vast majority of biological catalysts are protein enzymes.
Ribozymes. Okay, noted. But mostly proteins.
Mostly proteins. And their defining characteristic is their incredible catalytic power. They make biochemical reactions happen at astonishingly high rates, millions or even billions of times faster than the same reaction would occur without the enzyme. They are biological accelerators. It's useful to contrast them with inorganic catalysts like platinum or nickel used in industrial chemistry. Those often work best at very high temperatures and pressures. Enzymes, being proteins, are generally much more sensitive. Most enzymes get damaged or denatured by heat, typically above 40–50 degrees C for human enzymes, because that heat disrupts their delicate tertiary structure, including the active site.
Right, heat breaks them down, like cooking an egg.
Exactly like cooking an egg. Once denatured, the activity is usually lost permanently. However, there's an interesting exception relevant for biotechnology and exams: enzymes from thermophilic organisms. These are microbes that thrive in extremely hot environments like volcanic hot springs or deep-sea hydrothermal vents.
Thermophiles, heat lovers.
Right. Their enzymes are naturally adapted to be stable and functional at very high temperatures, sometimes even 80–90 degrees C or higher. This thermal stability is a remarkable adaptation. Now, to understand how enzymes work, we need a basic grasp of chemical reactions and their rates. Remember, a physical change just alters the state or form, like ice melting, without breaking chemical bonds. A chemical reaction, however, involves breaking existing bonds and forming new ones, creating new substances, like starch being broken down hydrolyzed into glucose molecules.
Physical versus chemical change. Got it.
The rate of a reaction is simply how fast it happens, usually measured as the amount of product formed per unit time. If we consider direction, we might call it velocity. And rates are influenced by factors like temperature. You might remember the rule of thumb from chemistry.
Rate doubles for every 10-degree rise, something like that.
Exactly. Roughly doubles or halves for every 10 degrees C change in temperature. Within limits, of course. Now, enzymes completely change the game regarding rates. Let's take that amazing example of carbonic anhydrase. This enzyme is found in our red blood cells and other tissues. It catalyzes the reaction. CO2 plus H2O, H2CO3, carbonic acid. This is vital for CO2 transport.
Carbonic anhydrase, okay.
Without the enzyme, this reaction is incredibly slow. Maybe only 200 molecules of carbonic acid are formed per hour, very sluggish.
200 per hour.
Okay.
Now, add carbonic anhydrase. How many molecules are formed? About 600,000 molecules per second.
Per second. 600,000. That's insane.
It is. The enzyme accelerates the reaction rate by about 10 million times. That illustrates the sheer power of enzyme catalysis. And our cells contain thousands of different enzymes, each specialized for a particular reaction or a small set of related reactions. Often reactions occur in sequences, where the product of one enzyme becomes the substrate for the next. Such a sequence of enzyme-catalyzed steps is called a metabolic pathway.
Metabolic pathway, like a production line.
Exactly like a biological production line. For example, breaking down glucose to get energy, glycolysis, involves a pathway of 10 distinct enzyme-catalyzed steps to convert one molecule of glucose into 2 molecules of pyruvic acid. And depending on conditions like oxygen availability or the organism, like yeast, subsequent enzyme steps can lead to different end products like lactic acid or ethanol. Enzymes provide this controlled, efficient, and flexible chemistry essential for life. So the big question, how do enzymes achieve these incredible rates? This brings us directly to that concept you mentioned, activation energy. Let's tackle this carefully.
Okay, activation energy. Ready?
In any reaction, the starting molecule is the substrate, S, and it gets converted into the product, P. So SEP enzymes facilitate this conversion.
Substrate to product, S to P.
The first absolutely necessary step is that the substrate S must bind to a specific region on the enzyme called the active site. Remember that pocket or crevice in the tertiary structure?
And the active site.
The substrate fits into this active site. much like a key fits into a lock, though the fit isn't perfectly rigid, as we'll see. This binding forms a temporary complex called the enzyme-substrate ES complex.
Enzyme plus substrate makes ES complex.
Okay. This ES complex is transient. It doesn't last long. While the substrate is bound in the active site, the enzyme puts stress on its bonds or brings reacting groups together, making it easier for the reaction to occur. The substrate is contorted into a highly unstable, high-energy state called the transition state structure.
Transition state.
Yeah.
Like the top of a hill.
Exactly like the top of a hill. Imagine a graph plotting energy against the reaction progress. The substrate, S, is in an energy valley. The product, P, is in another energy valley, which might be lower or higher than S, depending on the reaction. To get from S to P, the molecule must pass over an energy hill. The peak of that hill is the transition state.
Okay, got to get over the hill.
Right. The difference in energy between the starting substrate S and the peak of that hill, the transition state, is called the activation energy, EA. It's the energy barrier that must be overcome for the reaction to happen. Even if the overall reaction releases energy, P is lower than S, you still need to put in this activation energy to get it started.
Like needing a match to start a fire, even though fire releases heat?
Perfect analogy. You need that initial input of energy. Now, here's what enzymes do. They do not change the starting energy of S or the final energy of P. The overall energy change of the reaction remains the same. What enzymes do is dramatically lower the activation energy barrier. They lower the height of that hill.
They make the hill smaller.
Precisely. They provide an alternative reaction pathway that requires much less activation energy. By lowering the barrier, they make it much, much easier and therefore much, much faster for substrate molecules to reach the transition state and become products. That's the secret to their speed. Let's quickly summarize the catalytic cycle in steps. This is often asked about. 1. Binding. First, the substrate S binds to the enzyme's E, active site, specific fit E plus S, E, ES.
Step 1. Binding. 2. Induced fit. Often the binding of the substrate causes the enzyme to change its shape slightly, creating an even tighter, more optimal fit around the substrate. This is called the induced fit model. The enzyme adjusts to the substrate. Step 2. Induced fit, like a handshake adjusting. 3. Kinalysis. Now within the active site, the enzyme facilitates the chemical change. Bonds in the substrate are broken and your new bonds are formed, converting S into P while still bound to the enzyme. This forms a temporary enzyme product, EP complex, ES. Step 3. Catalysis, making the product. 4. Release. Finally, the enzyme releases the product, is P. The enzyme E itself is unchanged by the reaction and is now free to bind another substrate molecule and start the cycle all over again. EP, please P. Step 4. Release. And the enzyme is ready to go again.
Exactly. Ready to go again, potentially millions of times per minute. Okay, so enzymes are powerful catalysts, but their activity is very sensitive. Let's look at the key factors affecting enzyme activity. Changes in these factors can alter the enzyme's tertiary structure, especially the active site, and thus affect its catalytic efficiency. These are prime targets for exam questions.
Factors affecting activity.
Right.
Like temperature.
Temperature and pH. Absolutely. These are critical. Enzymes typically work best within a very narrow range of temperature and pH. There's an optimum temperature and an optimum pH where the enzyme shows its maximum activity.
Optimum conditions.
Yes. If you go below or above this optimum range, the activity usually declines sharply. Think of a bell-shaped curve on a graph plotting activity versus temperature or pH.
Bell curve, okay.
Now, low temperature, like a refrigeration or freezing, generally just slows the enzyme down and makes it temporarily inactive. Its structure isn't usually permanently damaged. If you warm it back up to the optimum, it often regains activity.
Low temp, inactive, but recoverable.
Right. But high temperature, significantly above the optimum, is different. It causes denaturation. The heat energy makes the enzyme molecule vibrate so much that the weak bonds holding the tertiary structure together break. The protein unfolds, the active site loses its specific shape, and the enzyme loses its activity permanently. Like cooking that egg, you can't uncook it.
High temp denaturation, permanent damage.
Got it. Similarly, extreme changes in pH, too acidic or too alkaline relative to the optimum, can also alter the ionization state of amino acid R groups involved in maintaining structure or in the active site itself, leading to denaturation and loss of activity. Most human enzymes have an optimum pH around neutral, 7.0, 7.4, but some, like pepsin in the stomach, work best at very low pH, around 2. 2. Concentration of substrate. This is another important factor, often shown graphically. What happens if you keep the enzyme concentration constant and gradually increase the amount of substrate? The reaction gets faster. More substrate means more product. Initially, yes. As you increase substrate concentration S, the velocity rate of the reaction increases. More substrate molecules are available to collide with and bind to the enzyme's active sites.
Makes sense.
But this increase doesn't go on forever. Eventually, the reaction rate reaches a plateau. It hits a maximum velocity, which we call Vmax. Beyond this point, even if you add more and more substrate, the reaction rate does not increase any further.
It levels off. Why?
Because at Vmax, the enzyme is working at its full capacity. All the available active sites on all the enzyme molecules are occupied or saturated with substrate molecules. The enzyme molecules are processing substrate as fast as they possibly can. Adding more substrate doesn't help because there are no free active sites left to bind it. The enzyme concentration itself becomes the limiting factor.
The enzyme is saturated, like all checkout counters being busy in a store.
Exactly. Opening more counters, adding more enzyme, would increase the overall rate, but adding more customers' substrate won't help if all counters are already occupied. Now, there's a value related to this curve called Crillem, the Michaelis constant.
Kaminow, I've seen that.
Kms is defined as the substrate concentration S, at which the reaction velocity is exactly half of Vmax, Vmax2. Kelement is a measure of the enzyme's affinity for its substrate. A low kelem means the enzyme has a high affinity. It can reach half its maximum speed, even at low substrate concentrations. A high kelem means low affinity. It needs a lot more substrate to get up to speed.
Low kelement equals high affinity. High kelement, low affinity. Okay, that's important.
3. Inhibitors. Enzyme activity can also be affected, specifically reduced or stopped, by certain chemical substances called inhibitors. The process is called inhibition.
Inhibitors slow down or stop enzymes.
Yes. There are different types, but one very important type for exams is competitive inhibition. A competitive inhibitor is a molecule that closely resembles the enzyme's normal substrate in its shape and chemical structure.
Looks like the substrate.
Exactly. Because it looks similar, it can bind to the enzyme's active site, the same place the real substrate binds. It essentially competes with the substrate for access to the active site.
Competes for the active site.
Okay. So, if the inhibitor molecule binds, the real substrate cannot bind. As a result, the enzyme's overall activity decreases because fewer enzyme molecules are available to process the actual substrate.
Makes sense. The inhibitor blocks the site.
A classic example is the inhibition of the enzyme succinic dehydrogenase, involved in cellular respiration, by a molecule called malonate. Malonate looks very similar to the enzyme's real substrate, succinate. So malonate competes with succinate for the active site and inhibits the enzyme. This principle of competitive inhibition is actually used medically. Many drugs work by inhibiting specific enzymes, for instance, in bacteria, to stop infections.
Using inhibitors as drugs. Interesting.
Okay, just a brief mention of classification and nomenclature of enzymes. With thousands of enzymes known, we need a system. They are classified into 6 major classes based on the type of reaction they catalyze. Each class has subclasses, and each enzyme gets a unique 4-digit number, the EC number. You don't need to memorize all the numbers, but knowing the 6 major classes is important.
6 main classes. Okay.
1. Oxidoreductases, dehydrogenases. Catalyze oxidation reduction, create redox reactions, transfer of electrons or H atoms. Think energy metabolism.
Oxidoreductases redox.
2. Transferases. Catalyze the transfer of a specific chemical group, like an amino group, methyl group, phosphate group, but not hydrogen, from one molecule to another.
Transferases transfer group.
3. Hydrolases. Catalyze hydrolysis reactions, breaking bonds by adding water. Think digestion, breaking down proteins, carbs, fats.
Hydrolases break bonds with water.
4. Lyases. Catalyze the removal of groups from substrates, but not by hydrolysis, often forming double bonds or rings, or the reverse reaction.
Lyases remove groups, not water, often make double bonds.
Isomerases. Catalyze the rearrangement of atoms within a single molecule, converting it into an isomer.
Isomerases rearrange atoms, make isomer.
6. Ligases. Catalyze the joining together of 2 molecules, forming new bonds like CO, CS, CN. This usually requires energy, often from ATP hydrolysis. Think synthesis reactions.
Ligases join molecules together, use energy, got the 6 classes.
Finally, one last crucial aspect: cofactors. Many enzymes don't work alone. They require an additional non-protein chemical component called a cofactor to be active. The protein part of such an enzyme, which is inactive without its cofactor, is called the apoenzyme.
Apoenzyme, protein part plus cofactor, non-protein part, active enzyme.
Exactly. Apoenzyme plus cofactor equals holoenzyme, the complete active enzyme. There are 3 main types of cofactors. 1, prosthetic groups. These are organic compounds, and they are tightly bound, often covalently, to the apoenzyme. They are essentially a permanent part of the enzyme's structure. Example: HAM is the prosthetic group in enzymes like peroxidase and catalase, which break down hydrogen peroxide. The HAM group is essential for their function.
Prosthetic groups organic, tightly bound, like HAM. 2. Coenzymes. These are also organic compounds, but their association with the apoenzyme is transient, meaning it's temporary. They bind only during the catalytic reaction, often acting as carriers of specific chemical groups or electrons between reactions. Coenzymes can often work with multiple different enzymes, an extremely important point for nutrition and exams. Many vitamins are essential components of coenzymes. For example, the vitamin niacin, vitamin B3, is part of the coenzymes NAD and NADP, which are crucial electron carriers in metabolism. Coenzymes organic, loosely bound, often derived from vitamins like NAD. 3. Metal ions. Many enzymes require one or more inorganic metal ions for activity. These metal ions can help bind the substrate, participate directly in the catalysis, or stabilize the enzyme structure. They form coordination bonds with amino acid side chains in the active site and sometimes with the substrate itself. Example, zinc, Zn++, is a crucial metal ion cofactor for the digestive enzyme carboxypeptidase. Metal ions inorganic help binding or catalysis, like zinc.
The bottom line is, if the cofactor is removed from an enzyme that requires one, the enzyme loses its catalytic activity. This highlights just how crucial these cofactors are for enzyme function. Phew! We've really covered a lot of ground today. It was indeed a deep dive into the world of biomolecules, a topic that's absolutely fundamental to understanding life and, of course, crucial for your exams.
Definitely a lot, but really helpful.
So just to summarize the key takeaways. Living organisms, despite their diversity, share a fundamental chemical basis. While they use the same elements as non-living matter, the relative abundance is key, especially the high proportion of carbon and hydrogen.
Right, the CNH abundance.
We learned how scientists analyze tissues separating into the acid-soluble pool, micromolecules like amino acids, sugars, nucleotides, and the acid-insoluble fraction, macromolecules like proteins, polysaccharides, nucleic acids. And importantly, we clarified why lipids, despite being small, end up in that insoluble fraction due to their role in membranes and behavior during extraction.
The lipid exception, yeah, got that now.
We explored the amazing world of proteins, their 4 levels of structure: primary, secondary, tertiary, quaternary, and how this hierarchy dictates their diverse functions as enzymes, hormones, antibodies, transporters, and structural elements. We also looked at polysaccharides as energy stores: starch, glycogen, and structural components, cellulose, chitin, and nucleic acids, DNA, RNA, as the carriers of genetic information.
And macromolecules covered.
And then critically, we dove into enzymes. We broke down how they work by lowering activation energy, walked through the catalytic cycle step by step, and examined the crucial factors that affect their activity, temperature, pH, substrate concentration, and inhibitors, including the mechanism of competitive inhibition. We also touched upon their classification and the essential role of cofactors like prosthetic groups, coenzymes, often involving vitamins, and metal ions. This knowledge isn't just for exams, it's the foundation of understanding all biological processes and even many medical treatments.
That was incredibly thorough. Everything feels much clearer now, especially the enzyme mechanism lowering the hill and the protein structures. Breaking it down step by step and using those analogies really helped visualize it all. Thank you so much.
I'm really glad to hear that. That's the goal. Now, for your upcoming exams, NEET, boards, whatever you're preparing for, I strongly recommend you revise this chapter again. Pay close attention to the specific examples given for each type of biomolecule, especially the secondary metabolites and protein functions. Really nail down the steps of enzyme action and how those factors affect the rate graphs.
Okay, will do. Review the examples and enzyme graphs.
And definitely practice past year questions specifically on biomolecules and enzyme kinetics. They are very frequently asked, so practice is key to recognizing the patterns and common pitfalls. And remember, if any doubts linger or pop up later as you revise this chapter, please don't hesitate to reach out. Keep up the hard work, keep exploring these fascinating molecules, and I'm confident you will ace your exams.